ಸರ್ ಸಾಗರ್ ಸರ್, ನಾ ಗಣೇಶ್ ಕೂಡ ಮಾತಾಡೋದು. ಸರ್ ತಮ್ಮಲ್ಲಿ ಶೇರ್ ಹಾಕಿದಿಲ್ಲ ಸರ್, ಪ್ರೆಸೆಂಟ್ನ ಒಂದು ಬರ್ತಾಯಿಲ್ಲ ಸರ್. ಸರ್ ತಮ್ಮಲ್ಲಿ ಪ್ರೆಸೆಂಟ್ನ ಒಂದು ಬರ್ತಾಯಿದೆ ಸರ್ ಅಲ್ಲಿ ಇದರಲ್ಲಿ ಸ್ಕ್ರೀನಲ್ಲಿ. ಹಾಂ ಹಾಂ ಸರ್, ಓಕೆ. ಗೊತ್ತು ಸರ್, ತಮ್ಮೆಲ್ಲ ಪರಿಚಯ ಇದೆ ಸರ್, ಆರಾಮಾಗಿದೆ ಸರ್. ಇಲ್ಲ ಸರ್, ಇಲ್ಲ ಸರ್, ಹಾಂ ಪ್ಲೀಸ್ ನೀವು. ಹಾಂ ಸರ್, ಹಾಂ ಓಕೆ. ಗೌಡ ನಾಯಕ್ ಗೊತ್ತಾಗ್ತದೆ, ಹೋಗಿ ಒಂದು ಮಾತಾಡಿ ಇರ್ತಾರೆ. ಯಪ್ಪು ಅದೇಂಗೆ ಹಾಂ ಸೀನಿಯರ್ಗಳನ್ನ ಹಂಗೆ ಹೋಗಿ ಅವನೇ ಮಾತಾಡಿ. ಶೇರ್ ಮಾತಾಡೋಕ್ಕೆ ಸ್ಪೀಕರ್ಗೆ ಏನು ಕೇಳಿದೆ ನಾವು ಮೆಟೀರಿಯಲ್ ಕೊಡಿ ಅಂತ ಪ್ರಾಬ್ಲಮ್ ಓದ್ಕೊಂಡ್ಬಿಡ್ತಾರೆ ಅಂತ. ಸಸ್ಪೆನ್ಸ್ ಏನಾಗಿಲ್ಲ ಅಂತ ಹಂಗಿಲ್ಲ.
Sir, slides are visible, sir?
ಇಲ್ಲ ಸರ್, ಬಂದಿಲ್ಲ ಅಂತ
Sir?
ಸರ್ ಜಾಸ್ತಿ
ಸರ್
ಸಾಗರ್ ಸರ್ ಇನ್ನೂ ಕಾಣ್ತಿಲ್ಲ ಸರ್.
ಕಾಣಿಸ್ತಾ ಇಲ್ವಾ ಸರ್?
ಕಾಣ್ತಿಲ್ಲ.
Maybe now. Now?
ನಾಟ್ ಯೆಟ್ ಸರ್.
ನಾಟ್ ಯೆಟ್ ಸರ್.
ಪ್ರೆಸೆಂಟ್
ನಾವು ಅನ್ಬೇಕು ಸರ್ ಅಂತೆ.
ಹಾ ಪ್ರೆಸೆಂಟ್ ನಾವು ಅಲ್ಲೇ ಸರ್.
ಪ್ರೆಸೆಂಟ್ ನಾವು ಕ್ಲಿಕ್ ಮಾಡಿ ಶೇರ್ ಸ್ಕ್ರೀನ್ ಅಂತ ಹೇಳ್ರಿ ಸರ್.
ಅಲ್ಲಿ ಎರಡು ಬರ್ತಾ ಇದೆ ಸರ್, ಶೇರ್ ಸ್ಕ್ರೀನ್ ಅಲ್ಲಿ.
ಶೇರ್ ಎಂಟರ್ ಸ್ಕ್ರೀನ್ ಅಂತ ಮಾಡಿ ಸರ್.
ಶೇರ್ ಎಂಟೈರ್ ಸ್ಕ್ರೀನ್ ಅಂತ ಮಾಡಿ ಸರ್.
ಓಕೆ, ನಂದು ಡಿಸೇಬಲ್ ಆಯ್ತು ಸರ್. ಇದು ಪ್ರೆಸೆಂಟ್ ನೌದು
ಇಲ್ಲ ಸರ್. ಇಲ್ಲ
ಅವ್ರು
ಡಿಸೇಬಲ್ ಆಯ್ತು
ಸರ್
ಸರ್ ಡಿಸೇಬಲ್ ಆಗಿ ನೀವು ಮಿನಿಮೈಸ್ ಮಾಡಿ ಸರ್ ಬರ್ತಾ ಇದೆ ಎಲ್ಲ
ಈಗ
ಇಲ್ಲ ಸರ್ ಇನ್ನು ಕಾಣ್ತಿಲ್ಲ
ಇಲ್ಲ ಇಲ್ಲ ಏನು ಕಾಣ್ತಿಲ್ಲ ಸರ್.
ಜಸ್ಟ್ ಎ ಮಿನಿಟ್ ಐ ವಿಲ್ ಲಾಗೌಟ್ ಆಗಿ ಲಾಗಿನ್ ಆಗ್ತೀನಿ ಸರ್.
ಸಾಗರ್ ಸರ್
ಸರ್ ಲಾಗೌಟ್
ಲಾಗೌಟ್ ಆಗಬೇಡಿ ಸರ್, ಯಾಕಂದ್ರೆ ಯು ವಿಲ್ ನಾಟ್ ಬಿ ಏಬಲ್ ಟು ಲಾಗಿನ್ ಅಗೇನ್ ಬಿಕಾಸ್ ರಶ್ ಐತೆ ಈಗ.
ಓಕೆ
ಸೊ ಯು ಟ್ರೈ ಜಸ್ಟ್ ದೇರ್, ಡೋಂಟ್ ಲಾಗ್ ಔಟ್ ಪ್ಲೀಸ್.
ಕ್ಲಿಕ್ ಮಾಡಿರಿ ಎಂಟರ್ ಸ್ಕ್ರೀನ್ ಅದು ಬರ್ತದೆ ಬಂದಿದೆ ಬಟ್ ಅವರು ಇದೆಲ್ಲ ಬಂದಿದೆ ಅವ್ರಿಗೆ ಆಪ್ಷನ್ ಬಂದೈತೆ ಅವ್ರಿಗೆ ನೋಡಿಲ್ಲ.
ನೆಟ್ವರ್ಕ್ ಪ್ರಾಬ್ಲಮ್ ಇದೆ
ನೆಟ್ವರ್ಕ್ ಇಶ್ಯೂಸ್ ಇದೆ. ಸಾಗರ್ ಸರ್ ನೀವು ಪ್ರೆಸೆಂಟ್ ಅಂತಂದಾಗ ಪ್ರೆಸೆಂಟ್ ನಾವು ಅಂತಾಗಿ ಇವರ ಎಂಟರ್ ಸ್ಕ್ರೀನ್ ಬರ್ತಾ ಇಲ್ಲ.
ಸಾಗರ್ ಸರ್
ಸರ್ ಪ್ರೆಸೆಂಟ್ ನಾವು ಡಿಸೇಬಲ್ ಆಗಿದೆ ಸರ್. ಲಾಗೌಟ್ ಆಗಿದ್ಮೇಲೆ
ಆದ್ರೆ ಲಾಗೌಟ್ ಆಗಿದೆ ಜಸ್ಟ್ ಹಾಗೆ ಇಲ್ಲಿ ಯಾರು ತೆಗಿತೀವಿ ಅಬ್ದುಲ್ ನರಫ್ ಯಾರು ಅಂಬಿಕಾ ಸಿ ಮತ್ತೆ ಯಾರು ವೇಟಿಂಗ್ ಇತ್ತು ಬಾರ್ ಕಷ್ಟ.
लेफ्ट साइड कॉर्नर में देखिए हुआ हो सकता है ब्लॉक करा हुआ है लैपटॉप का लेफ्ट साइड कॉर्नर में देखिए इट मे बी ब्लॉक्ड ऑन लेफ्ट साइड कॉर्नर. लेफ्ट साइड कॉर्नर में मे बी ब्लॉक्ड, you must unblocked and then you will be share the screen.
ಸಾಗರ್ ಯಾರನ್ನು ಗೊತ್ತಾಗಲ್ರು.
ಸಾಗರ್ ಡಿ ಅಂತ
ಬಂತ ಸರ್ ಒಳಗೆ ಬಂದ್ರೆ
ಇಲ್ಲಿ ಬಂದಿಲ್ಲ,
ಲಾಗೌಟ್ ಆಗಿಲ್ಲ, ಇನ್ನೂ ಲಾಗೌಟ್ ಆಗಿಲ್ಲ.
ಇಲ್ಲ ಸುಬ್ರಹ್ಮಣ್ಯ ಸರ್ ಬಂದ್ವಿ ಮತ್ತೆ
ಬಂದ್ರೆ ಸರ್
ಸಾಗರ್ ಲಾಗೌಟ್ ಆಗಿಲ್ಲ
ಸರ್. ಪಿ ಪಿ ಟಿ ಕ್ಲಾಸ್ ಅಂತ ಹೇಳ್ಬಿಟ್ಟು ನಡೀತಿಲ್ಲ ಲಾಸ್ಟ್ ಸಾಗರ್.
ಯುವರ್ ಎಂಟೈರ್ ಸ್ಕ್ರೀನ್.
ಹಾಂ ಎಂಟೈರ್ ಸ್ಕ್ರೀನ್ ಅಂತ ಎಸ್ ಸರ್. ಶೇರ್ ಆಗ್ತಿದೆಯಲ್ಲ ಅಂತ ಏನು ಕೇಳಿ ವೇಟಿಂಗ್ ಮಾಡೋಣ, ಯಾರು ತೆಗೆದ್ರು ಪಟ್ನೆ ಬಂದೇ ಬಿಡ್ತೀನಿ ಸರ್. ಸರ್ ತೆಗಿಯೋಕ್ಕಾಗ್ತಿಲ್ಲ ಸರ್, ಶೇರ್ ಆಗ್ತಿಲ್ಲ ಸರ್, ಶೇರ್ ಸರ್ ಶೇರ್ ಆಗತ್ತಿಲ್ಲ ಸರ್. ಹಾಂ ಹಾಂ ಏನು ಕಾಣಿಸ್ತಿಲ್ಲ ಸರ್, ಏನು ಕಾಣಿಸ್ತಿಲ್ಲ. ಅದೆಲ್ಲ ನಮ್ಗೆ ಶೇರ್ ಮಾಡ್ತೀವಿ ಸರ್, ಮೇಲ್ ಕಳಿಸ್ಬಿಡ್ತೀರಿ, ನಾವೆಲ್ಲಿ ತೆಗಿತೀವಿ, ನಾವೆಲ್ಲಿ ತೋರಿಸ್ತೀವಿ ಅಂತ ಇಮೇಲ್ ಹಾಕಿಬಿಡ್ತೀರಿ ಸರ್. ಕಾಂಪ್ರೆಸ್ ಮಾಡಿ ಇಮೇಲ್ ಹಾಕ್ತೀರಿ ಸರ್, ಇಮೇಲ್. ಹಾಂ ಹಾಂ ಅಷ್ಟು ಮಾಡಿಬಿಡಿ ಸರ್. ಅದೇ ಮೇಲೆ ಪ್ರೆಸೆಂಟ್ ನಾವು ಅಂತೇಳಿ ವೆಂಟರ್ ಸ್ಕ್ರೀನ್ ಕೊಟ್ಟಾಗ ಎಲ್ಲ ಕಾಣುತ್ತೆ ಕ್ಲಿಯರಾಗಿ. ಇದು ನಮ್ಮದು ಎಲ್ಲ ಪ್ರೆಸೆಂಟ್ ಆಗ್ತಾರೆ. ಹ್ಞೂ ಅಂತ ಹಾಗೆ ಮಾಡಬೇಕಾಗತ್ತೆ, ಮಾಡ್ತೀನಿ ಸರ್. ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡೋಷ್ಟರಲ್ಲಿ ಬಂದುಬಿಡುತ್ತೆ ಅವ್ರ ಮೇಲೆ ಅಟ್ಯಾಚ್ ಆಗಿ ಸೆಂಡ್ ಮಾಡಿದ್ರೆ ಇಲ್ಲ
ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿ ಗ್ಯಾಪ್ ಮಾಡೋದು.
ಎಲ್ಲ ನೀವು ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತೀರಾ ಸರ್, ನಾವು ಡೌನ್ಲೋಡ್ ಮಾಡ್ಕೊಂಡು ಈ ಕಡೆ ಬಂದು ತಕ್ಷಣ ಮಾಡ್ತೀವಿ ಆನ್ಲೈನ್ ಹೀಗೆ. ಓಕೆ
Ganesh, I just sent you by
Okay, sir.
mail.
Okay, sir. We are checking. Sir.
Yes, sir.
We are checking. Sir, we are checking.
Sir, we are checking, sir.
We are checking.
We are checking.
ಕಳಿಸಿದರೆ ಕಳಿಸಿದರೆ ಇನ್ಬಾಕ್ಸ್ ಬರುತ್ತೆ, ರಿಫ್ರೆಶ್ ಕೊಟ್ಟು ಸೆಂಡ್ ಆಯ್ತು
Sagar
Sir, send ಆಯ್ತಾ ಅಲ್ಲಿಂದ?
Sir, send. Sir, you sent us.
Just I shared by email.
Okay, sir.
Okay, sir. Okay.
Those
ನಾಟ್ ಇಟ್ ರಿಸೀವ್.
to SK Chitral.
Okay, sir.
Okay, sir. Okay, I'll check this. I'll check.
Yes. Ganesh, I just sent you by
Okay.
mail.
Okay, sir. We are checking. Yes, we are checking. Sir, we are checking.
Sir, we are checking, sir.
We are checking.
We are checking.
ಬಂದಿದೆ ಸಂಜೆ.
Sagar, sir, send ಆಯ್ತಾ ಅಲ್ಲಿಂದ?
Sir, send. Sir, you sent us.
Just I shared by email.
Okay, sir. Okay.
Those
Send to
to SK Chitral.
Okay, sir.
Okay, sir. Okay, I'll check this. Sharing ಅಲ್ಲೇ ಬರ್ತದಾ? ಇಲ್ಲಿ ಬರಲ್ಲ.
ಹೆಸರು ಕಳಿಸಿ ರೀಸೆಂಟ್ ಮಾಡಿ.
ಇದು ತೊಲಿಬನಿ ಇದರದ್ದು. ರಿಲೀಫ್ ಮೇಲೂ ಸ್ವಲ್ಪ ಹೆಡೇಕ್ ಆಗುತ್ತೆ.
Got it?
Yeah,
I've received, sir. I'm just downloading it, sir.
Later, sir. ಬಂತು ಸರ್. ಓಕೆ.
Start only.
Yes.
ಡೌನ್ಲೋಡ್ ಮಾಡಲಿ. ಓಕೆ, ಸೇವ್ ಲಾಗೌಟ್ ಮಾಡಿಬಿಟ್ರಿ. ಎಲ್ಲ ಬರ್ತಲ್ಲ, ಇದು ಬರಲ್ಲ. ಶೇರ್ ಮಾಡ್ತೀರಿ? ಶೇರ್ ಮಾಡು, ನಾ ಇದು ಮಾಡ್ತೀನಿ, ಇಂಟ್ರಡ್ಯೂಸ್ ಮಾಡ್ತೀನಿ ಅಷ್ಟೇ. ರಿಲೀಫ್ ಮೇಲೂ ಸ್ವಲ್ಪ ಹೇಗಿದಲ್ಲ ಸರ್.
Got it?
Yeah, I've received, sir. I'm just downloading. Okay. So can you see this slide, sir?
Yeah.
Okay, sir. Okay. Sorry, sir. There is some network issue, it seems. So that's why you are getting the.
Not from first slide. Just
Okay.
second slide is showing.
Okay.
Yeah.
Okay.
There was some technical issue.
There was some technical issue. First of all, I welcome Dr. Subramanian, sir. So, Principal Scientist, Division of Entomology, Indian Agriculture Institute, New Delhi. And I also welcome all the participants.
Okay. I also welcome all the participants to the program.
I also welcome all the participants
Today's
to the program. And today's session is going to be engaged by Subramanian, sir. And the topic actually is the characterization of the gut microbes.
Of the symbionts in the insects.
Of the symbionts in the insects. Okay, and this is the topic, and this is going to be engaged by
Principal
Dr.
Scientist,
S.
Division of
Subramanian,
Entomology,
the Principal Scientist at Division of Entomology, Indian Agriculture Research Institute, which is popularly known as, abbreviated as IARI, and it is New Delhi. Okay, and Subramanian sir has completed his postdoctoral studies from the Oxford University and did his PhD from the Indian Agriculture Research Institute, New Delhi, and completed his post-graduation from the Entomology University. And Subramanian sir has the several professional recognitions to his study. And the first and the foremost topic is the fellow of Royal Entomological Society, London, and research advisor for the Nang Academy of Science, Singapore, and fellow of Entomological Society of India, and chairman to the editorial advisory committee, Indian Journal of Entomology, and associate editor to the Allopathy Journal. And apart from that, the Subramanian sir has the professional experience in teaching, research and extension as well, and expertise in the characterization of the gut microflora of the insects in from 2014 to 2015, and is the number of gene bank submissions on the 16S rDNA genes of the gut bacterial isolates from the insects, 400, and the postdoctoral training on the molecular characterization of the gut microflora in the insects at Oxford University during the year 2009. And current research interests of Dr. Subramanian sir, they are the molecular characterization of the phosphate resistance in insects, and the molecular characterization of the gut microbes and symbionts in insects. And Subramanian sir has the academic experience of teaching the students and guiding the undergraduate and the postgraduate, and even the doctoral students also, right from 2004 to, and it is still on. And he has several research papers, publications to his credit, and he has as many as 75 research papers, and he has edited 3 books and 12 book chapters and popular
articles of 25 in number, and he has attended totally 76 of the scientific conferences and the seminars and abstracts. And apart from that, Subramanian sir has the most valuable research projects also to his credit. And the first and the foremost of it is the biological traits of Asian genetic groups of whitefly initiated by the endosymbionts and endophytes. And this was funded by Department of Science and Technology, and this is still on, and it is up to the 2022. And deploying the biotechnological based decision making tools for the post-market pest management to enhance the security and the to market access. And this is the project which has been funded by Department of Biotechnology and AIF. And there is another project which has handled the molecular characterization of the resistance to the insecticides and the fumigants in the stored product insects in the northeastern regions. And this was funded by GPT again. And apart from that, he has several research papers to his credit, and they are ranging from the topmost is the 11.13 NAS rating, and which is really practically impossible for any ordinary scientist or any researcher for that matter. And this is an illustrious career, so that the Subramanian sir has come through. And now I would request Dr. Subramanian sir to take charge and deliver the lecture on today's lecture. Please, sir, can you hear me, sir?
Okay, thank you for the profuse compliments. Just I'll proceed as such you can move the slides, isn't it?
Sure.
I'm glad to be associated with the 10 days national training. Next slide. Today's lecture is on the structural and functional character. Next slide, please. Or if you can give the command, I can move from here. It's not there here.
Nice. Can you see that, sir?
Slide. Second slide, the structural and functional characterization of insect gut bacteria, the endosymbionts. Can you see that?
Yeah.
Okay.
Yeah, I could say.
Okay, sir.
Okay.
Okay, this lecture I'll give an overview of the insect gut bacteria and endosymbionts. This I made it as a two-part series. First, I will tell about the significance of the insect gut bacteria endosymbionts. Second one, how we can do this, how we are doing the structural characterization, and how we are going about the functional characterization. Just let us see an overview of insect gut bacteria and endosymbionts. Next slide, please. First thing is, we want to know who are they, because the first and foremost of the analysis of the gut bacteria, the endosymbionts are, what are the microbes that are associated with the insects. Because if you take the gut microbiome, it's a huge number. Maybe around 10 power 9 to 10 power 13 microbes are there, maybe in a normal lepidoptera and caterpillar, and the counts may vary, even if it is in to the order of 10 power 20 in insects like termites, where they are solely depending on the microbes for many of their functional characterizations. So it is important to know how and what are the insects, what are the microbes that they can associated, for which we normally go, there are certain terminologies. Microbiome, it tells about the whole lot of the microbes associated with any ecosystem. It may be the soil microbiome, or it may be the gut microbiome, or it may be the ocean microbiome. So microbiome is a very holistic terminology, tells about the holistic association of microbe associated with the entire ecosystem. Here with respect to insect gut, how we are assessing the microbiome. Foremost important is the diversity analysis. It can be done by the different techniques. Either we can go for the culturable isolates, or second one is we can go for them using the genomic tools. So for example, we have taken studies wherein we have taken comparative analysis of the gut bacteria, the three different lepidopteran insects. Just Just a minute, Ganesh. I will share the another PowerPoint. I think this is the second one has come first.
Okay, sir. Okay.
Let me share the other slide wherein I can give you the first slide.
Okay, sir. Okay.
First introduction part, sorry.
Ganesh.phd@gmail.com, sir.
Next prize in the characterization of
Sir, please
microbes.
send the mail to
ganeshpatil.phd 2014-2015.
Okay, Ganesh Patil.
.phd@gmail.com, sir. So that I can
download
Okay.
bacterial isolates from the insects 400 and the postdoctoral training on the molecular characterization of the gut microflora in the insects at Oxford University during the year 2009. And current research interests of Dr. Subramanian sir, they are the molecular characterization of the phosphate resistance in insects and the molecular characterization of the gut microbes and symbionts in insects. And Subramanian sir has the academic experience of teaching the students and guiding the undergraduate and the postgraduate and even the doctoral students also right from 2004 to history on. And he has several the research papers, publications to his credit, and he has as many as 75 research papers, and he has edited 3
Okay, Ganesh, I shared the file.
chapters and popular articles of 25 number and 76 scientific and apart from that, Subramanian sir has the most valuable research the projects also to his credit, and the first and the foremost topic is the biological traits of Asian genetic groups of whitefly, the vegetative basil, the service endosymbionts, and endophytes. And this was funded
Sorry
by
for the inconvenience.
Technology. And this
Actually, I could not open it here.
And it is up to the 2022. And deploying the biotechnological based decision-making tools for the post-harvest Insect Pest Management to enhance the food security and the market access. And this is the project which has been funded by Department of Biotechnology.
Sir, I have not received it. I'm waiting for this.
Project which has handled the molecular characterization of the resistance to insecticides and fungicants in a stored product insects in the northeastern regions. And this was funded by GPT again. And apart from that, he has several.
Yes, sir. Now I got it. Sir.
His credit.
Just I'm downloading, sir.
Yeah.
Just in the 11.13 NAS rating.
Okay, I to check it's a part 1, it should come first.
Yes.
Scientist or any researcher for that matter. And this is an illustrious career so that the Subramanian sir has come through. And now I would request Dr. Subramanian sir to take charge and deliver the lecture on the
second and question to first one. Sir, this is the.
Yeah, next. Yeah. First, let us see scope and prospects of the insect endosymbionts and gut microbes. What is their special significance? What are their role in insects? This is a first part pre-introduction. Next slide, please. So if you take the insects, you could see the diversity of the insects. Insects live in the different ecosystem, and one of the important advantage for the adaptation of the insects is they could able to cope up with exploit the ecosystem, and the foremost evolutionary features that is helping them is their gut physiology. The anatomy of the physiology in such a way it will suit the ecosystem. If you take it, if it is sucking insects, the intestine is adapted in such a way it will be facilitating the processing of the liquid diet. Similarly, in the case of the chewing insects like Dictyopterans, it will be a well-developed one with the gizzard and other organisms for the chewing of the hard materials. In the case of termites and others, there will be specialized pouches will be there, so that they will be helping them. So this anatomy of the intestine or the evolutionary of the gut, that is a foremost important feature for harboring the diversity of the gut microbes. Next slide, please. So how we can assess the gut microbes? There are two approaches. Generally, one is a culture dependent one. In earlier times, we used to isolate the microbes with the help of a number of microbiological media. So the power of the resolution or detection is with the power of the isolation techniques only. Earlier, whatever the media we have, that is enough for isolating the limited number of microbes. With the availability of the molecular tools now, even without isolating the cultures, we can identify what are all the microbes associated with the insects or in any organism, for which there are specialized molecular techniques are used. One such technique is use of 16S ribosomal DNA primer. During the course of the evolution of microbes, especially in the bacteria, the 16S subunit ribosomal DNA has been conserved one. So this DNA conserved nature have been exploited for isolating for detecting the microbes with a molecular fingerprinting. So we will be designing the specific primers in such a way it can detect the presence of a eubacteria or the bacterial genome. And here again, for each microbe, the 16S rDNA sequences region will be unique, based on which the species identity can be seen. The other aspect is the denaturing gradient gel electrophoresis. This is also one technique by which we can know, or when there are different stages these microbes are present, what are the polymorphism present. Based on the polymorphism value, the DGG can help to associate the diversity in one ecosystem to other. The foremost or recent technique is the next generation sequencing technique. The entire genome will be sequenced. So the small bits of genomes will be taken up, and with the help of the assembly and processing techniques, we will be identifying the different microbes through which these genomes have been originated. Earlier it was only with the help of microbiologists we could do the characterization. Now with the help of these genomic techniques and the molecular approaches, it is fairly easy for any biotechnologists or molecular biologists for the characterization of gut microbes. Next. So it's a complex community of microorganisms that live in the digestive tract, that what we call gut microbiota or the gut flora. So insects are colonized by the diverse group of the organisms. They may be the yeast, or maybe the bacteria, or fungi, or viruses, and they have their interspecific and intraspecific relationship with them, and that help them in many different ways for association with the insects. Next. Next slide, please. So it's actually a multifarious community that will be harboring the digestive tract. Some of them are maybe beneficial to them, that we call them as symbionts. How they are actually beneficial to the insects? So they may be helping them in the food digestion, or they may be helping them in the nutrition, or degradation of a fungal toxin, or helping in synthesis of pheromone products, or helping them to maintain the pH in the insects in such a way other digestive processes are facilitated. In some cases, the microbes provide a condition in such a way they will provide the thermal tolerance or resistance to the parasite development, or when the insects are exposed to the insecticides, they will detoxify the insecticides so that the insect environment will be harmless. So in these different ways, the microbe help the insects, and when these are beneficial to the insect, they are called as symbionts. Next slide, please. What are the different factors that will influence the colonization? Number one is the gut pH. It is a foremost important. In different insects, the pH range varies. Accordingly, only those group of microbes which are adapted to the particular level can grow on them. Second one is the oxygen levels. When the oxygen levels are full, aerobics can grow or aerogens can grow. In some cases, the insects have certain anaerobic chambers where they will harbor only the anaerobic microbes, and they do the special function. So whether it is oxygen dependent one, they are called aerogens, and there is no access for oxygen, or if it is anaerobe means they will facilitate only the growth of anaerobes. Similarly, the redox potential of the gut system as well as the nutrient availability, these are also the other factors that will be actually determining the different colonization of the microbes. Next slide, please. First, we have to understand what are you. Just whenever we go to a person, we want to know what are you doing, and we also should know who are you. That means we want to know who is the person, what is the name of him. So in the same, the study of microbes also, first we should understand the identity of the microbe. Next one, what it is doing inside the insect, what is their functional role. So these variations may occur insect to insect. In certain times, it may be there different biotypes. In certain cases, it varies with the same insects in different populations, it may vary. Because of these variations, these microbes give the adaptive advantage to the populations. There are certain specific situations, they will enhance the detoxification of the foreign material. So, to the specific condition only, we can isolate them. Similarly, in some of these polyphagous insects, that they will confer the host plant plasticity. So, in some cases, monophagous, they will be associated only with the limited host plant. So, the host plant association is also helpful in determining the microbial diversity. Next slide, please. There are a wide range of group of bacteria associated with the insects. Commonly, the gamma proteobacteria, alpha proteobacteria, firmicutes, or these are the other groups which I have listed in this slide. They are associated with the insects. This association with a different phylum or the group varies with the group of insects. Because it may not present in all insects, it may varies with a particular group of insects. It all depends on their evolutionary adaptation. Second one, in a particular insects, a particular community of the microbes will reside. So they will determine what is the usefulness to the insects. So if you see the case of termites, only a specific community of microbes will be there, which are specializing on decomposing the wood or detritus material. Similarly, you could say different association with the honeybees or bumblebees. So depending upon the group of insects, the community of bacteria or the microbe associated will be different. Next slide, please. So just to show you for your information, one of our studies were carried out wherein we assess the diversity of the microbes in the insect diamond back moth. So these are the assorted microbes we could see. Next slide, please. So this is another study wherein two biotypes, B biotype and Q biotype, have been taken up, and within the same insect also, between the biotypes, there are certain variation in the microbial association. So this community association varies between insect to insect or even within the insect between different biotypes or the populations. Next. Similarly, there are certain specialized situations wherein only specialized microbes can grow. So there will be specialized niches in the intestine. Here there is a one slide showing there are microbes that will be attached to lactobacilli group. This is commonly present in the honeybee, and there are specialized niches in the intestine of the honeybee wherein this lactobacilli will be profusely grown. So insects should provide a special niches for this community of the microbes that are helpful to them or beneficial to them. Next slide. So in some cases, say for example, disease condition. If you say the healthy and diseased one, there may be the variations. The difference in variations may also helpful whether they can allow the pathogenesis of a particular disease causing microorganisms or not. Next slide. So these are all about the gut bacteria. Next terminology is the endosymbionts. Now there may be the distinct variations. All are bacteria or the microbes associated with the insects, but there are we have a different terminologies. When they are colonized in the gut, they are called as a gut bacteria. In some cases, insects house them in a specialized cells. Insects have in the fat bodies a particular type of cells called the bacteriocytes. These bacteriocytes have provided some kind of housing facility to the specialized, these are the organelles for housing the microbes. Say for example, in the case of hemipteran insects, they don't have the ability to have amino acid synthesis or the protein synthesis. They will be passively feeding on the phloem fluid, which are rich in sugars only. So during the course of the evolutions, many of these hemipteran insects have given a niche for certain microbes, or these are called as an endosymbionts. They have even provided the housing facilities to them, just in the way we want to greet our guests. When some guests are coming, we will be providing a special guest room. Similarly, for VIP guests, we will be providing the guest houses. In the same way, insects provide certain housing facility for some of these microbes which are beneficial to them. These microbes will help them in improving the suboptimal diet, because the phloem fluid of the hemipteran insects will be lacking the protein. So they will be needing the help of the microbes to synthesize the amino acids and protein, for which the insects and microbes have a symbiosis. If the insects provide a specialized cells for housing, and if the microbes are residing in them, they are called as an endosymbionts. These endosymbionts, it is a kind of a remarkable evolutionary adaptation, the intimate association of the particular microbe with a particular group of insects. The study about the endosymbionts has started as early as 1854 by Leidig. Leidig is one of the pioneer insect microbiologists who have profound out the concept of endosymbionts in insects. And many path-breaking studies have been taken up during the course of the studies of endosymbionts, like workers like Blockman or Buckner. With the help of their studies and the recent molecular techniques, helps to characterize these endosymbionts, what are their role. To functionally distinguish them, there are different types of classifications are there. One classification is primary and secondary. The other classification is obligate endosymbiont, another one is a facultative endosymbiont. Obligate endosymbiont means these are the group of endosymbionts which will be present in all members of the insects. Say for example, in the case of Bemisia tabaci, irrespective of the genetic group variations or the population variations, this Portira will be present. This Portira is one of these obligate endosymbiont which helps them in the amino acid synthesis. When Portira is absent means the survival of the insect will be in the question. So these are called as an obligate endosymbiont. Without this Portira, the Bemisia tabaci cannot survive. Second category is facultative endosymbionts. These are the endosymbionts. They may be present or may not be present in all the members of the populations. Whitefly have the association of different secondary endosymbionts. Say for example, Cordinium, Rickettsia, Wolbachia, Fritzia, there are different groups are there. The variation here is, say there are 100 insects, in all 100 insects Portira will be there, but not Cordinium. In some population Cordinium may be present as 10%, in some there may be 0 or some may be even 100. Similarly with the Rickettsia, similarly with the Wolbachia. So these variations assign them as the status of facultative endosymbionts, but their role is also important because they will be giving certain adaptive advantages to them. While the primary endosymbiont benefit them nutritionally, the reproductive endosymbionts like Wolbachia, they manipulate the host reproduction. For example, Wolbachia is one such endosymbiont. They are known as a male killers. During the stage of cytogenesis, they will kill the male embryo and leave the insects only female. In certain group of insects in Hymenopterans, the Wolbachia presence is very much important to make the insects teletoki. They will produce only the teletokous ones in the presence of Wolbachia because they will kill all the male embryos and they will facilitate only the feminization of the insects and they will give them adaptive advantage. Similarly, thermal tolerance. When there is a heat stress, there are certain microbes like Rickettsia that aid them to tolerate the excess heat. Similarly, in some cases, some of these microbes, they resist the parasitism by the other endoparasitoids. Or in some cases, they will resist the entomopathogenic organisms. So by this way, these secondary endosymbionts, they provide either the adaptive biological traits or fitness traits. They improve the fecundity or their reproductive capability. By all means, these primary and secondary endosymbionts facilitate the life of our insects in a better way. Next. So just I will give you some of these classic examples, how insects ensures the intimate associations of the endosymbionts. One example is the stingbug species, Megacopter punctatissima. Here, the particular microbe Ishikavella capsulata, they will reside in the gut lumen. Here, there is one specific case. They are not associated in them in a specialized cells like bacteria sites. Rather, when the insects are there during the development stages or in the nymph and adult stages, this particular microbe will be reside in the gut lumen. But during the stage of reproduction, they will be transmitted through the eggs, and these endosymbionts will be transmitted in a form of the capsule, and this will be carried forward to the next generation. Next slide, please. So here, when the insects lay the egg, along with them, the ovipositing females defecate to produce a specialized endosymbiotic capsule on the outside of the egg case. You could say here, this is actually the egg case. So they will produce a small capsule on the outside of the egg case. So immediately after the hatching, the juveniles which are coming out, they will be feeding on these capsules and they will acquire the endosymbionts. This particular study proves even endosymbionts not necessarily can be housed inside, but insects have certain specialized adaptation by which they will ensure the progenies also would be getting the same endosymbionts. So this is one of the classic case. It's called as a capsule endosymbiont. Initially they will be present in the gut of the insects, but during the reproductive stage they will be coated or smeared on the eggs. Next slide. There is another insect, Riptortus pedestres. In this particular case, they have a specific endosymbiont called Burgholdera. This endosymbiont is not present in the gut of the insect at all, or not they will be ensuring the vertical transmission, but rather this Burgholdera will be present in the environment or the ecosystem where the bugs are there. They will acquire from that ecosystem, and every generation the insects will acquire the endosymbionts, and they have specialized crypts inside the midgut where the Burgholdera will be colonizing. Hosts that fail to acquire the endosymbiont during the specific developmental stages may actually display the stunted growth. So this is again ensures the intimate association of the endosymbionts either through the parents or through the environment. Next slide, please. Now we can see, first we have seen about how the niches are present. I have not present the whole cases. It may be vary with the Excel because some of them I will be showing the second part of the slide. In the case of termites, they will be having a specialized organ called a paunch, where the microbes will be there present in a specialized chamber. Similarly, in the case of white grubs, they have a adaptation called a fermentation chamber, where they will be harboring the microbes or anaerobes that will in turn help them to digest the humus and other organic matter. So likewise, endosymbionts are those ones which have been provided intimate cells, or they may find ways for the intimate association with the insects. Now let us see some of the functional role of the endosymbionts. The foremost important one is the nutritional symbiosis. Say for example, in many cases of the insects which will be feeding on the wood or hard cellulose material, the endogenous cellulases are not sufficient to digest the cellulosic materials. Most of the time the cellulases, these are the enzymes, cellulases, lipases, amylases, pectinases are the enzymes complemented by the microbes. In the case of honeybee also, they identified one particular endosymbiont, Giliamella apicola. This particular endosymbiont helps them in the pectin degradation. When the insects collect the pollen and nectar, they will be stored in the honey stomach. This particular microbe will be there in the crop of the honeybees, and they will help them in the degradation of the pectin. Pectin is one of the hardest layer to digest. So the pollen, outer coat of the pollen will be with the pectin, and the pectinases by this endosymbiont helps them to digest the pollen, and with the mixing of this pollen and nectar in the honey stomach, they will be preparing the honey. Similarly, many different organisms, many different ways of enzyme complementation will be there, and they provide the nutritional symbiosis to the insects. Next slide, please. Now, there are techniques by which we can identify whether it is a cellulases provided by the endogenous insects or the cellulases provided by the microbe. The recent studies have shown wherever the cellulases complemented from the microbe, they will be having the cellulose binding domain. So, the insects which will be feeding on the wood or the cellulosic material, we can very easily identify whether it is provided by the cellulases enzymes secreted within the insect or the cellulases complemented by the microbe by analyzing the CBM moiety of the cellulases. By this study, many insects it has been proved that the cellulases have been derived from the microbes. Next slide, please. So there are different enzymes are acting because cellulose digestion is a complex process. So there may be the enzymes like endo beta 1,4 gluconases or extra beta 1,4 cellohydrolases or beta glucosidases. Likewise, all the complementing enzymes by the synergistic action with the endogenous cellulose, they will be acting at the different levels of the cellulose chain, releasing either the cellobiase or the glucose. So a complex process of digestion has taken place, and this is one of the classic cases, the cellulose degradation. They essentially require the intimate association with the different groups of microbes. Some microbes may be having the endogluconase activity, some may be in the cellobiase activity, some may be having the glucosidase activity. Complementing all, the consortium of the microbe, they help in the digestion of the cellulose and release the glucose at the end. Next slide. So this is one of the nutritional endosymbiont we have seen. Similarly, there are many other features like insect-plant interactions, the endosymbionts will be helping. Insect-vector interaction or insect natural enemy interaction. We will see one or two case studies. Next slide, please. Insect defense. In the case of the insects, there will be the particular microbe that will be colonizing inside the intestine. This colony of the microbes will not allow the colonization of other pathogenic microbe. They will provide the growth conditions in such a way very difficult for the pathogenic organism to colonize as long as the symbiotic bacteria in good number. So this is what is called the colonization resistance. Same principle only, same thing have been adapted for human being also. Whenever there is a gastroenteritis or any other intestine infection, the intestinal microbe will be depleted. So due to the course of antibiotic we are consuming. So what the normal physician suggesting is they will be giving a dose of probiotics. Probiotics are nothing but the consortium of beneficial microbes. Because initially these colonization they will be resisting and they might have been lost during the process. So they will be supplementing again the symbiotic microbes in the form of a probiotics. Same thing happening with the insects also. They will be providing as a foremost resistance for the colonization. In European bumblebee, they have found out whenever there is a good colony of the symbiotic microbes, they will resist the pathogen of a particular trypanosomid, Crithidia bombay. Similarly, there are many case studies proved when the gut microbes in good number, European foulbrood infestation is not prevalent at the wide scale. This particular aspect is called as a colonization resistance. The second aspect is the intestinal microbes or the beneficial microbes, they will synthesize the antibacterial compounds. For example, in the case of honeybee, we have proved that we isolated one of the actinomyces, Streptomyces norsei. It has the capability of producing an antibiotic. This antibiotic was having the good antifungal activity against the entomopathogens pathogens, Beauveria bassiana and Metarhizium anisopleae. So by the production of antibiotics also, these gut microbes will degrade the pathogenic organisms. So and many studies have proved that by improving the gut bacteria, the pathogenesis or the pathogenic microbes growth can be curtailed in beneficial insects. Next slide, please. So this is the holistic one. Either it may be the colonization resistance, or maybe the toxin breakdown, or nutrient supplementation by the enhancing the digestive activity, or certain times they will the degradation product, they will be part of the aggregation pheromones. So a complex of activities or the beneficial activities are conferred by the microbes and symbionts. Next slide, please. There are some cases wherein gut microbes are associated with the insect plant interactions. In the case of Acetosyphon pisum, they found out a particular protein is COO2 in the salivary gland, and when they are knocking down, actually their host searching ability is stopped. They found out this actually derived from the microbes. Next one. Again, there is another interesting story. In the case of whenever there is a high concentration of the bacterial endosymbiont, Candida silurus, there will be the reduced expression of the plant defense. Whenever the insects are feeding, the plants also having the separate defense pathways, like there are different defense expression genes will be there, or the defense system of the plants will be activated so that herbivory will be arrested. To overcome this, some of the psyllids, they will be having the certain microbes, they will suppress the defense operated by the plants, defense system operated by the plants. They proved the same thing with the Diabrotica virgifera, a major pest of maize. Whenever they have the colony of the Wolbachia, the defense expression of the plants will be suppressed. Next slide, please. Similar which are shown that gut bacteria or the gut endosymbionts, they will provide a protective coating. The groil is one of the chaperone protein or a carrier protein. Some of the viruses, they will be covered by the groil protein of the endosymbionts. Only when they are covered by the endosymbionts, they will be free from the immune response action of the insects, and this groil protein of the endosymbionts is very much important for the virus transmission. This is a study taken up in India only in the lab of Dr. Rajagopal Raman and others. They have proved that the groil protein from the endosymbiont Arsenophonus is very much critical, and it interacts with the coat protein of the viruses, and this coat protein and the interaction with this groil protein is important for the transmission of cotton leaf curl virus in cotton, or the tomato leaf curl virus in tomato. So by this way, virus, vector and symbiont interactions are also have been recorded widely. Next slide, please. There is another classic case. In the case of grapes, there is a disease caused by a bacteria. It's called as a bacteria Pierce disease. So by manipulating of the certain endosymbionts, there is a bug called a sharpshooting bug. What they did actually was, by manipulating the endosymbionts of the bug, they could be able to prevent this plant pathogenic disease in the case of the grapes. So this approach is called as a paratransgenesis. The microbes can be manipulated, and the released microbes can help the insects or help the plants for the prevention of the diseases. So the microbes as such can be used for, the symbiotic microbes can be used for the pest control as well. This is called as approach called paratransgenesis. Next slide, please. How this paratransgenesis or the manipulation of symbionts can be used? There are many different case studies are there, especially with human vectors. There are proven cases are there. Number one is the Chagas disease. This Chagas disease is caused by the Trypanosome cruzi. How they did was in the Rhodnius prolixus, they manipulate the gut endosymbionts in such a way, whenever this modified gut endosymbionts are there, they will prevent the proliferation of the trypanosome. What they did was, this modified gut microbes, they release it in the environment because Rhodnius prolixus, these bugs have the habit of feeding the fecal matter of the insects. So these gut microbes have been camouflaged in such a way as a fecal matter of this, and they release into the environment. And whenever they take up these modified microbes, and they will prevent the carrier of the trypanosoma, and thereby the Chagas disease can be prevented. Is in the control of dengue, they use the Wolbachia. Wolbachia, I said, there is one reproductive endosymbiont. This reproductive endosymbiont can be triggered for cytoplasmic incompatibility. So between the populations, only when compatible Wolbachia will be present, there will be mating success will be there. They manipulate the endosymbionts of the mosquito population with the two different strains of the Wolbachia, both positive and negative strain. So whenever there is an alternate strain is introduced in the population, there will be the cytoplasmic incompatibility or the reproductive failure will be there, by which the multiplication of the insects would be stopped, thereby the transmission of dengue can be prevented. Similarly, it has also been proved in the malaria control also, the manipulation of the gut microbes have been taken up. Some of these cases like manipulation of the Wolbachia have been taken up in a large scale. Australia and Queensland University, they have successfully demonstrated this technology, and this technique developed by Queensland University have been adopted by the Brazil for the control of dengue through preventing the multiplication of Aedes aegypti. This is one of this approach, like genetically modified organism. They are not the genetic modified organism. Rather, the introduced microbes will be released into the environment, which will be harboring only in the insects, and it will not enter into the human system. By this way, the disease-causing vectors can be controlled. This is one of the tremendous advantage in the manipulation of the endosymbionts for the welfare of human health. Next slide, please. Similarly, in Africa, there is a particular disease, river blindness. Just click next. Here again, one particular endosymbiont Wolbachia have been associated, and simply by giving the antibiotic drug, earlier normally they will be taking the anti-helminthin tablets for controlling the river blindness. And by taking this simple antibiotic, the Wolbachia can be controlled. When the Wolbachia is controlled, as such, the carrier of the filarial worms will lose the ability for transmitting the river blindness disease. So this is one of the successful protocol where the endosymbiont suppression can helpful. Similarly, a Melinda Gates Foundation funded project is operating in the Bihar and other regions for controlling the Kala Azar through the manipulation of endosymbiont. Especially they are working on manipulation of the Wolbachia in the population sandflies, by which the Kala Azar disease can be controlled. Next. So by which way the paratransgenesis have been worldwide, it is approved for control of many of the vector-borne diseases. Even in India also it has been, trials have been taken up or at the early stage of the evaluation of the paratransgenic approach for the control of Kala Azar. Next. Just I want to highlight some of the applications, because many of the applications said they have the specialized abilities. So these specialized abilities can be used for human welfare as well, especially some of the lactobacilli group which are helpful for human can also be manipulated through the honey, and there are already some probiotic honey have been developed in US, wherein whatever be the probiotic microbes can be delivered through the honey bees. And other approach is cellulases, especially in paper industry, pulp industry, wherever this cellulose degradation is important. The insect derived cellulose are one of the potential targets for the quick degradation of the materials, or even biocomposting also. The insect borne cellulosic or pectidolytic activity microbes offer a huge potential for converting the biomass into ethanol, or plant biomass can be degraded in a quick way. They have identified in one of these books lipase enzyme technologies. Many of the lipases derived from insects and other microbial organisms. They are the potential beneficial prospects for different industries like detergents, food or dairy products, cosmetics, tea processing, leather processing, etc. You could notice in some of the detergent, you could find granules. These granules are nothing but the lipases. Those are the enzyme granules which will be having the lipases activity, which will be removing the dirt from the cloth. So in many of the detergent formulations, they use the lipases from different microbial source. Here again, insect derived lipases have a lot of potential for developing or enhancing the ability of the detergent. Similarly, for coloring matter in food or dairy products or cosmetics, oil processing, or many different biopolymer, there are ample scope for exploiting the insect microbes for the industrial uses as well. Next slide. So I just conclude this slide the first part of my presentation with all these. So as there is a wide diversity of the insects, there will be greater adaptation of the insects and their anatomy of the gut, and that gives the way for the diversity of the gut microbe. This diversity of the gut microbe help them in a different way for the insect benefit, as well as same thing can be exploited for the human benefits as well. Next slide, please. So this is actually the take-home message. Trust your gut. So the gut bacteria have a lot of scope and prospects for their nutritional advantage to the insects, and same can be harnessed for pest control as well as for the human welfare. That's all, Ganesh, for now.
Okay.
And I have the next slide presentation. I think I can give some 5 minutes break to them.
Okay, fine.
In between, if they have any specific such questions or clarification for the first part, they can ask.
Yes,
Yes.
So now
Because the first part I have covered only the what are the scope and prospect, basic terminologies.
Okay.
Second part I will tell them how we have to do the structural and functional characterization with the case studies.
The topic is
open for discussion now.
Are there any
Are there any
Or if
specific
not, we
questions?
can have the question at the end of the session.
That should be good,
That should be good, sir. That should be okay, sir. That should be fine.
It should be okay. Now let them take some 5 minutes break.
Okay.
Okay.
It's also monotonous for them also. I will also take a cup of water in between.
Yeah, you can.
Yeah, you can, sir, of course, but it's very interesting, in fact.
Second part Second part start ಆಗಿಲ್ಲ.
ಸ್ಟಾರ್ಟ್ ಹಾಗೆ ಸ್ವಲ್ಪ ಆಗಿತ್ತು ಸರ್ ಅಷ್ಟೇ, ಒಂದು ಸ್ವಲ್ಪ ಇಷ್ಟು ಮಾಡಿದರು.
Subramanian sir, there are no doubts from the audience side. May I ask you questions regarding this particular segment that you have taken up now?
Ganesh, you can keep the previous slide.
Yes, sir.
Go to the second, yes. Just when you are ready, you can tell, I can start.
Sir,
How long the session is?
It can go up to any longer, sir. But we have commenced at 3 o'clock. Generally, it will be 5 o'clock. But there is no problem, sir. Time is not the limit.
Anyway, I'll also try to complete within the time limits. There was a hiccup at the beginning because of the screen sharing.
That's why we
have not kept the end limit for that. So you can proceed. Sir, I have a doubt. In fact,
Yes, please. Yes.
Don't mind. So you talked about the transfer transmission of the microbes.
Microbes and symbionts.
Yeah. Because they are not intracellular, and they actually culture is made on the eggs, you said, isn't it?
Yes.
What exactly will do the larvae? Will it ingest or what once it comes out of the egg?
Ingest. Many larvae have the habit of ingesting the. As soon as they emerge from the eggshell, there will be contents of the egg, or sometimes they will be having the habit of licking the eggshell. So during the licking, they will be acquiring. Whereas in the case of capsule symbionts, they will chew as well as suck the saps of this.
Okay.
From the capsule, like a chewing gum, a small capsule will be there. They will be chewing and sucking from the capsule. They will be getting the microbes.
Okay. One more thing, since it is this particular field of entomology which is developed to such an extent, so why is that not very well exploited in the case of providing the resistance to pesticides in the honeybees? Because the honeybees, the major constraint in the pollination or the cultivation, rearing the beekeeping itself is the pesticides. So why is that the culture which can confer the resistance against many pesticides can be colonized? It is not that easy as much as we speak. But even as it tried.
No, sadly we have not progressed well in the honeybees as such. Even in gut microbes also only very limited workers are working.
Yes.
I have been working there consistently for about 13 years in this field. So there are very few workers are consistently working on the gut microbes, and hardly we find workers in the field of apiculture also consistently on this, because it cannot be done overnight with a single isolation or so. We have to study a whole lot of population over the years to come to a conclusions. In the case of European consortium and others, they have very big projects on the honeybee gut microbiome projects, internationally funded ones, covering in many different European countries. They come with a number of probiotic supplements for the growth of, for helping the honey yield as well as for even resisting the diseases. So likewise, we have to have more number of projects. We just only started working on the honeybees in the recent years. So it may be possible, potential is there.
Okay.
Can I come in? Can I come in for a minute, sir?
Yes, please.
This is Dr. Nisar speaking from Sukast, Kashmir, sir.
Yes, welcome.
Thank you for your wonderful presentation. I have a small bit of query regarding this transoral transmission of diseases.
Yes.
Sir, I am working in the Department of Sericulture here. We have a problem in silkworms, that is the protozoan disease pebrine.
Yeah, pebrine. Yeah.
Which is caused by highly virulent protozoa Nosema bombaceus.
Protozoan, yeah.
We have been trying a lot to take control of this protozoa, but the results are not that encouraging.
Yeah.
It's a problem over here. So, can you please let me know what could be the most effective methodology to take care of this protozoa under the temperate conditions?
What about this disease-free laying? Do you follow the protocol? There is a normal one they are suggesting for protozoans.
So exactly. We are going for.
Isn't it?
Yeah.
I also been working in sericulture for about 6 years, where they normally increase the mother moth examination and elimination at the egg laying stage itself.
Yes, sir. We do follow that mother moth examination in later and separate. Now, what happens in our case? We'll get the disease-free layings, and suddenly in the third or the fourth age, we get the disease again and again.
Maybe there may be some That's what I can say. There may be some inoculum in the lot, egg lot or somewhere. So we should find the sources for the egg lot and we have to remove. As such, I don't have any specific ready-made answers with the gut microbiome for the protozoan.
Sir, one more small submission is there. If we go by the terminology of the disease, the silkworms should not, if it is the transovarial transmission, they should not progress beyond the third age. But they are going very quite nicely, and they are settling the cocoons as well, even if they have the disease. So, regarding this trend, whether it is Nosema bombaceus or it's NIK1, NIK2, so if sir, my request is if you can provide us the platform, we could enter into a collaborative project for that.
Yes, we can explore. One or two times I come to which campus you are there in Kashmir?
I am in Coast Kashmir, Sher Kashmir University of Agriculture, Science and Technology of Kashmir, Srinagar.
Which campus? Which campus? I went to Shalimar campus twice.
No, sir. I am at Mirgud campus, which is dealing with the sericulture.
Okay, how far from Shalimar that is?
It is just at the periphery of the Srinagar city. If we go from Lal Chowk, it is just 10 kilometers.
Okay.
But it's very much. But it is very much within the vicinity of the Srinagar city.
Okay, we can explore the possibility of studies there. Sure to contact you. Okay. Hope you.
Thank you very much, sir.
Next time when I come over there, or if you come over there,
I'll be.
Okay.
Yes, sir. I will be mailing you, sir. I will be mailing you. I'll be in touch with you, sir. Thank you. Thank you.
Yeah. Thank you.
Thank you.
Hello. Hello.
Yes, please.
Please.
Can I ask you some few doubts?
Of course.
Yes, sir. Yes, welcome. Who is? May I know?
Sir, this is Dr. Shiona from US Bangalore GKVK.
Yes, welcome, sir. Thank you.
Sir, thanks for nice presentation. Indeed, I have a few questions.
Yes.
The one is, I think in the 2017 there is one paper in the PNAS.
Yeah.
That is the caterpillars' lack of resident gut bacteria. They don't have any resident gut microbes. There was one study. Can you comment on that? What, because when you look at the studies and all the while you are telling that there is symbionts in the inside gut, but however there are scientists who have proved that there is no any resident gut microbes. But what about the diet which eats away, like for example a leaf or the stem, that kind of microbes which are remained in the inside gut. So that was their opinion.
No, that's all. Because it's all the kind of hypothesis they are providing, kind of the evidences they are giving. This PNAS paper, that particular paper is one, they say there is no specific resident, but there are many studies it has proven, especially they have proven in case of Lepidoptera in insects. Even in Lepidoptera in insects, always there are many studies on the resident microbe. This is only an exceptional paper. We also sometimes come across certain queries when our papers are got for the review, and we beat the reviews with convincing answers. Because there is no resident microbe. Even in human being, we are having resident organisms. Most of the organisms during the course of the evolution, they have acquired the microbes.
Certainly.
And there are two ways. We have to have a classic difference. One is the one that is coming from the environment, that is the passing organism. But only there are certain passing organisms will find a niche inside the intestine, and they are called as only the resident microbes.
Right, sir. Thank you. And I have another question, sir.
Yes, please.
And usually you somewhere just mentioned that there is secondary symbionts.
Yes.
Can we expect this secondary symbiont converted to be as a primary symbiont? If it is so, what is the mechanism which they follow?
No, not conversion. It is only classic difference. One, they are essentially needed. They are called as a primary. Because we have seen, especially with the whitefly, we have conducted a large number of studies. We have found in all populations, not only our studies, worldwide they found Portira is the primary and obligate. And there are certain populations where Cardinia is altogether absent, or the Rickettsia is altogether absent. But it is not a permanent condition. Maybe absent for the particular population. Maybe in other population they may be present. So that is actually the classic distinction between the primary and secondary. Only the infection frequency that varies with the populations. That's all.
Yes, I agree, sir. But my worry is that can we expect this secondary symbiont turn to be a primary symbiont sometime? Can insect use that?
No, in say for example in Asia 2-1, there is one particular genetic group of the whitefly where we have found consistently cadenium is absent at all. So in some way there is a combination of these facultative endosymbionts are present in the group of population. So likewise only the organisms are operating.
Yes, sir.
How to convert means because Portira alone have been proven for their role in amino acid synthesis. So that's why it is relegated to the status of primary.
Yes, sir.
But not the other endosymbionts.
Right, sir. Thank you, sir.
Yes.
Let us after the finish the part B.
Yes.
Thank you, sir.
Thank you. Now let us go for the second part. The first part I have given the example about the prospects and the scope of the gut microbe, where we have given an overview of the gut bacteria, what are gut bacteria, how they are colonized, what are the conditions, and second case defining what are the endosymbionts and the category like primary, secondary, and some assorted cases, because this is a vast field. Every year we are getting thousands of publications. Only some assorted cases I have chosen to give an example for the beginners. Now second part I will be focusing on how we will be characterizing these gut bacteria or endosymbionts. What are the different approaches, how we will be doing. There are two parts is there. One is the structural characterization, second is the functional characterization. The structural is finding out what are all the members present. Say likewise in this particular Google Meet on this training program, if you click the button participants, we know who are the participants in this. Same way, if we assess the diversity of the microbes in a particular organism, we found out what are all the microbes associated with them. This diversity analysis, it is what is called or covering as the structural part of the gut bacteria or endosymbionts. Second part is the functional endosymbionts. If we could be able to characterize some of them, this microbe is doing this function, this is called as the functional characterization. So that's why there are two questions are normally asked. One is the what are they or who are they? Second, how are they? How good they are as a beneficial or the transient microbes. As Dr. Shivana asked earlier, because many are only coming from the environment. So these are called as a transient ones. Just they may be acquired from the environment, but they may not reside in a long term. Only which of them are the beneficial to them, they will reside in the organism and they do the functional role. Any problem, Ganesh?
Sir.
Slide is not coming in the presentation mode.
Yes, sir. Someone is sharing, sir.
Somebody has made as a host.
Sir,
That's what
someone is sharing apart from me. So please stop sharing.
someone is shared. That's what is because
So I'm requesting
others,
please don't share
your
screen.
So next terminology I want to, before going to the one, metagenomics. Meta, you know, metadata, meta means multiple or the many. If we isolate the DNA from an insect, it's not only the DNA of the organisms, it is the DNA of the many different organisms that are residing in the insects. If we do the characterization of the whole genome, it is called as the genomic analysis. If we do the genomic analysis of all the organisms associated, that is called as a metagenomic. When we are isolating an extract from an insect, it contains the insect's own tissues besides the microbial organisms that are residing them. If we isolate the DNA from the whole one, it will have the DNA of the both the insect as well as the thousands of microbes in them. And there is a sequencing procedure that is called as a next generation sequencing, that they do the analysis of the metagenome. That is why we normally come across the term metagenomic analysis of the gut bacteria or the endosymbionts. Here we have taken up one particular study wherein we tried to do a metagenomic analysis of three different groups representing insects. One as a monophagy, second one oligophagy insect we have taken, another one a polyphagy insect. For monophagy, we have taken silkworm, Muga silkworm. It is also limited oligophagy only, so we can label it as a monophagy. Then for oligophagy, we have taken insects like a diamond back moth that will be present only on the cruciferous plants. And third one is the polyphagous insects, Helicoverpa armigera. We have taken up a study by both culturable isolation or culture dependent on the metagenomic analysis to know the structural diversity of the microbes. Next slide, please. So as I said earlier, first one was a silkworm, second one is the oligophagus, which will be feeding exclusively on the cruciferous plants. Next slide. The third sample we have taken up is the Helicoverpa armigera, American bollworm or a cotton bollworm. It is a polyphagous, feed on solanaceous crops, legumes and assorted cereals also like maize, etc. So we have taken a 3 different group of insects and we have investigated a study to know the gut bacterial diversity using both approach, both the cultural as well as the culture dependent one. Culturable means we will be isolating microbes from these organisms and identifying them, what are they. Second one is we don't isolate the cultures. Whole DNA will be isolated and we will be processing through the metagenomic analysis. Next slide. So I have given 2 terminologies. One is a culture dependent one, another one is a culture independent one. So here the first process what we will be doing is we will be isolating the culture from the different insects. We will be taking the sample insects, dissecting the gut, taking out the gut tissues, homogenizing them, and from them we will be doing the staking on the plates. First we will be doing the general staking, then again by serial dilutions we will be plating on them, and when the countable colonies are there we will take them, and those colonies we will be isolating in a single colony isolation. We will stick them. Initially we will be doing the smearings, next one we will do the streakings. Then if we identify the single homogeneous or a pure culture isolate, these pure culture isolates will be streaked on the media, and from this again we will be taking up for the broth or growing on the specific broth media, and they will be taking on the DNA from them. And this DNA, by using the 16S rDNA primers, we will be conducting the polymerase chain reaction. There are many primers are there for the 16S rDNA. We use the primers like 24 as a forward one, 1492R as the reverse one of the 16S primer. And these primers then we will be getting an amplicon. There's this somewhere around 1400 to 500 kb. This is the 500 base pairs. These amplicons will be excising them or isolating this amplicon, and this amplicon will be sent for sequencing. So after sequencing we will be getting the sequences. So each sequence will be from one isolate. So this sequence we will be doing the blast analysis. There are separate bioinformatic analysis are there. Once we get the sequences for a particular microorganism, 16S or RNA primer, this amplicon when we are sequencing, we will do the blast analysis. By doing the blast analysis, there will be the NCBI database of different, that is a collection of thousands of 16S rDNA gene sequencing from different microbes will be there. So when we are matched with the NCBI blasting for the microbes, we can get the tentative identification. So this may be probably a Pseudomonas or maybe a Bacillus. Likewise, we will be identifying them. Here we are not using any morphological characterization or not. Purely we are based on the 16S, because this is having a 97% similarity with the base organism. When here also we follow the rules of similarity matches. When the similarity matches is above 97 or so, we can take it up as the representative of the species. When the similarity quotient is less than 95, we can say it may not be an exact species. Then we can tentatively indicate only as a Pseudomonas species. If it is less than 92, even genome we cannot identify. We can tell them as maybe the species unidentified. So this is the way by which we will be isolating as well as identifying the microbe using the culture dependent technique. Next one is culture independent technique. Please same slide please. Same. The next technique is, here we are not doing any culturable isolates, or we are not doing any isolation of culture at all. Directly we will be getting some insect, we will be isolating the DNA. There are a specific protocol, there are different platforms are there, Illumina, like there are many different platforms are there, by which this genomic DNA will be subjected, and it will do the sequencing of all the metagenomic organisms associated, and we will be using certain specific bioinformatics filters by which this processing bioinformatic analysis only look for the microbial derived sequences, and from which we will be identifying what are all the different organisms associated with. This approach is called as a metagenomic approach by using the NGS, next generation sequencing technique. So these are the overview of the both culturable and inculturable techniques in the study of gut microbes and symbionts. Next slide. So as I said, we have taken up 3 different insects. We have found out about 86 isolates from silkworm, another 46 from the Helicobacter armigera, and another Plutella xylostella we have taken up 37 isolates. We compared them the proportion, because this proportion varies with a group of insects, but irrespective of the group of all the Lepidopterans, these bacillus were the predominant ones. So we identify the association based on the grouping of this hygrome. This is what is called the diversity analysis, how much percentage of them constituted by the different group of bacteria. Next. So by blast analysis, we will be identifying immunity. Initially we had about 150 sequences. Then by removing the redundancy or similarity, we have found out 86 unique sequences, and these unique sequences we have submitted as a GenBank accessions. And based on the blast analysis, we have found out these are all the bacterial isolates associated with these insects. There are certain specific software also available. Based on the function, these microbes predominantly they are associated. So these are all the certain speculation only. These are the probable functions. These microbes can be associated. We can guess. Second one. Next. We did kind of different populations. We collected the Muga silkworm from the different regions of the northeastern India. And we just tried to find out what are all the similarity between the different regions. The excerpts of this have been published in a paper in the Insect Molecular Biology in the year 2018. How the diversity varies with the different locations. We have collected the Muga silkworm from about 5 to 6 different northeastern states, and what are all the common one, what are the consistent one across the regions, and what are the overlapping one between the regions. Likewise, we made the analysis and the interpretations. Next slide. We can also find out which are the predominant one. This is what is called a Whittaker rank analysis. When there are say 86 isolates, which is the most predominant among all. So based on the periodicity of occurrence, the Whittaker rank abundance plot will tell. So what is the number 1 bacteria, number 2, number 3, accordingly it will give the distribution based on this presence across the regions. Next. Similarly, we did the isolate from the Helicobacter armigera. So here again, we have characterized all the isolates. The GenBank sequences have been submitted with this accession number. Next. Similar, another study with the Plutella xylostella. These are the isolates and their probable functions. Next. So previous slide, please. Previous slide. So here we found out there are different isolates. They may be having a different variations. So these kind of isolates, we could find out some of the common occurrence of the gut bacteria across the species, or which of the common for them, or which of them unique for the particular insects, it can be identified. But if you do the more sampling only, we can make a good interpretation, because this Plutella xylostella we have taken only one population. If we have to make a particular study on the core microbiome, still we have to increase the sampling. We have to do the isolation of this Plutella xylostella across the region, and irrespective of the regions, if there are any specific microbes are present, and we can identify them as the core microbiome. Or otherwise, these are only sample studies, they will tell about the culturable isolates. The limitations for this approach is, we cannot isolate all the microbes that are resident in the gut of the insect, because many of the isolates cannot be cultured with the kind of media we are using. Because most of the microbes, more than 85% are non-culturable. They can reside only in the gut fluid of the insects. But this assay, culturable assay, can somehow give a lead about the predominant ones in the insect. Then next question is how to go for the assaying them. For which we go for the second approach called the culture independent approach. Next slide please. Here, as I said, this is actually the analysis called the metagenomic analysis. Here the process is, this is the systematic one. We will be accessing the gut, isolating the DNA only from the gut portion. Genomic DNA will be extracted, and this genomic DNA will be having certain specific requirements as for the sequencing procedure. So what is the strength of the DNA? It should be there. What would be the quantum required? It varies with the different sequencing platform. As per the requirement of the sequencing platform, we have to process or prepare the sample for the metagenomic analysis. Then first thing they will be doing is the quality check, whether our DNA is the homogeneous one, or are there protein contamination, or can we use them for the quality check. Only when our DNA passes through the quality check, they will go for the sequencing. Here again, they do the same technique only. They will be going for the amplicon sequencing. They will be preparing the clonal library, and they will be preparing the sequence. Here the process with them, as per the earlier one, we could do the sequencing, that is what is called a Sanger sequencing. End-to-end sequencing, we can get the sequencing for about 1400 bases. Whereas in the case of next generation sequencing, they don't do the full sequence of each organism. Rather, they will be, amplicons will be prepared, and sequencing for about 100 to 200 base pairs will be sequenced. So it is like making a small cuts. So like different vegetables like carrot, beetroot, beans, all that chopped inside a mixie, it will be chopped into small pieces, fragments. Again, these fragments will be taken out, and we will be doing the reassembling. Like the orange color pieces are carrot, the purple color ones are beetroot, and the green color ones are bean. Same thing, similar type of analysis here being done. After doing the amplicon sequencing, based on the tagging, they will be identifying this is a Pseudomonas, this is a Bacillus, by which they will categorically define what is the operational taxonomy unit. So operational taxonomy unit means these are purported to be this organism. Based on the core features, they will probably belong to this species or genus. It will be identified. That is what is called the OTU classification. Based on this, we can do the alpha diversity as well as the beta diversity of the microbes. Next slide. Here in this case, we have gone for the paired end sequencing. The platform we have used is the Illumina platform. This is the most common one. The amplicon region we have taken is the V3 region of the 16S. There are 9 regions of the ribosomal RNA, of which V2 and V3 are the regions providing the variability. So we have chosen for the amplicon sequencing the V3 region for all the 3 species. Next. So the library will be prepared, and they give this kind of a data. What are the total reads? What are the sequence? And as I said, the sequence will be around 150 to 200 only. These are the total data generated. Next slide, please. Based on the available OTUs, they will categorize for each group of insects. So this is the analysis that will give, these are the actinobacteria. Likewise, they will give the, at the phylum level, what is the distribution in different organism, or at the class level, what is the different proportion of organism present. Next. So this is one kind of taxonomic classification. It gives information about the proportional representation of the different groups of the microbes. Next. It is at the genus level. So what is the Streptococcus? How many? Predominant one is this organism, Candidatus solibacter. So likewise, they identify the different one. These are all incalculable one. Candidatus means it is a tentative name. So most of them are unknown category only. They identify what are the predominant genus in this different insects. One is the Antheria, another is a Plutola, another is a Helicoverpa metagenome. Next. So this assay can give only information about the structural diversity. But here, interestingly, we found out what is the proportion of the OTUs of the total reads and what are the predominant groups present in each organism, and they do the diversity analysis. Interestingly, the diversity analysis results revealed that Plutocella xylosella was having the more diverse than. Hypothesis may be, since Helicoverpa armigera is a polyphagous, so it may be having the diverse bacteria. But the results show Plutocella xylosella, the diversity is very high based on the diversity indices. The probable reason being, unlike the other organisms, the Plutocella xylosella have to encounter the glucosidases and other toxins in the cruciferous. Only those organisms that are capable of processing these toxicants, they are called as a specialists. All the aphids cannot thrive on the crucifers. Only certain aphids can thrive. Similarly, only certain organisms which are capable of detoxification of the cruciferous toxicants can grow on the Plutocella xylosella, can grow on the cruciferous plant. The similarly, Plutocella xylosella is one of these adaptive capabilities there, and it depends on the diverse group of microbes, and obviously that may be the one probable reason that the diversity of the bacteria associated is very high in the Plutocella xylosella. So this is the one assay that gives only fairly an information called what are the different groups that are present. And next, based on which we can go for the specific culturable techniques by isolating the specific group of the bacteria. But this information or this metagenomic and culturable analysis cannot give the any specific information what are their functional role, for which we have to go for some other technique by which we can functionally assay them. So this assay I just showed you to demonstrate what is the how we can assess the structural diversity. Next slide. Next, our question hypothesis is, what are the role of the gut microbiome? Are there any specific role played by them? So for which we have to specifically design an experiment. We have to functionally enrich the group of microbe in such a way that particular function can be extracted. Say for example, if you want to address the query for the thermal tolerant means, we have to set up an experimental condition in such a way, one is the heat exposed one, another one is the normal one, and if you do the analysis, then we can find out what are all the bacteria that are enriched under the heat cells. Similarly, for cold cells it can be done. Similarly, it can be done for the insecticide detoxification or many different functional roles. So we have to specifically design the experiment for the functional validation. Here in our case, we have taken up the case of cellulose digestion, and we want to characterize those microbes which are functionally useful for digestion of cellulose. Next. Here we have taken up Muga silkworm as a test insect, because Muga silkworm is the one as a wild silkworm. They will be feeding on the Bombycina trees, and Som and Sol trees are their host plants. During the final larval instars, they will be feeding on the plants or the leaves with the high cellulose content. So our hypothesis was, especially the final instar larvae may be having the associated with the cellulose degrading microbes. What we did was, we extracted the gut of the insect, we take the homogenate of them, and we provided them in a cellulose minimum media with the cellulose only as a substrate. We have taken up filter paper as a substrate, and we have allowed them for the digestion of this filter paper. During about 4 weeks, we could find the entire filter paper have been smashed and a mash have been prepared by the action of the cellulose digesting microbes. So this enriched part we have taken up for our metagenomic analysis to identify what are the microbes that are associated with the cellulose degradation. Next slide. Here, instead of taking the acid the gut, we have taken the enriched gut homogenate, and we have gone for this platform. Here we have gone for a technique called whole genome shotgun sequencing. Instead of metagenomic analysis with a 6-genus amplicon sequencing, we have gone for another technique called whole genome shotgun sequencing. It means entire genome of this will be prepared as using a shotgun technique, and small pieces will be prepared and they will be assembled and for functional association. Next slide. So we have gone for the ion torrent platform, wherein we did this analysis and use this again. We use the filters called with the help of, they will enrich only the bacterial prokaryotes filter, and other genomes will be filtered, and when they are analyzed, this is the assay. Specifically, they have found out here, alkaligenase is the one family associated with more than 56.8, and Achromobacter is one of the genus more than 48.2. So unlike the earlier structural diversity, here we have one comparison. Here we found that one particular genus is predominantly enriched. So this gives an inclination, this particular group of this bacteria may be associated with the cellulose digestion. Next slide please. We proved the same with the rank abundance, relative abundance using the Whittaker rank index. Next. And as I said earlier, we found that particular Achromobacter xyloxidans and Achromobacter pichardi. These are the two microbes that are predominantly present about 48% of the whole isolates. So here it gives a clue. Unlike the clumsy clue we are getting about in the structural one, here it is only a limited group of the microbes that are associated with the cellulose digestion, and these are the key organisms that are associated with the cellulose digestion, we presume. And we found out certain clues. The predominant genus may be Achromobacter. Next slide please. Our phylogenetic analysis also revealed the same thing. Next. There is another assay like cluster of orthologous groups. They will assign the functions to the different action, and here we found that energy metabolism or the carbohydrate or metabolism will be the most commonly enriched one during the COG analysis. Next. Similarly, there is another pathway called a KEGG analysis. So that will analyze for the different biochemical pathways it's enriched. Here again, we found there is a metabolism of carbohydrate, or the cellulose digestion is predominantly enriched in this metagenomic analysis. Next. Based on that, we specifically designed the media for isolating them for the cellulose degrading bacteria, and we interestingly found out that alkaligens fecal is similar to them, was there about in a good similarity, and all these are novel isolates because they are having only 83% similarity with available isolates, and these isolates give a predominant information, and these are the novel isolates that may be having the cellulolytic activity. Our next step was characterizing this, same slide, characterizing them. Previous slide only. We prepared these isolates either as a pure one or consortium. Then we prepared the probiotic formulation. Then these probiotic formulations we have evaluated under the field conditions and also for the field validation. Next. So this is a one particular study by which the specifically designed experiment can identify a functionally group of one. So we, based on this assay, we have found out a consortium of bacteria which are involved in the cellulose digestion, and this was prepared in a probiotic consortium, and our field studies also give a good indication this probiotic consortium, when it is prime, that nutrition of the silkworm is enhanced at the final instars. This study we conducted at the Jorat and other regions. Next. So these are the one of the study I had taken up earlier. Similarly, we had taken up one more, just I will be rushing up, I don't want to give a pure elaboration. We had taken up 4 or 5 different species of the white grub. We just do the anatomical identity where those gut microbes are resided. Next. So we did the analysis, what are the gut compartmentalization, are there any specific microbes in the different regions of the gut. Next. So we did them with more sampling, because that is what multiple sampling is required, more than by about 4 to 5 years we conducted from the same sampling area, so by which we can give the information of the core microbiome associated with the insect. Next. So initially we did it with the Lepidiota mansciata 1 white grub, then we conducted with Anomola dorsalis, then Anomola species. Next, one more study we conducted with Anomola dimidiata. So by having a comparative studies, we could find out the core microbiome associated with the white grub. So this also, as I said earlier, multiple sampling or similar species in the same environment, if we get and do the thorough analysis, we could identify the core gut microbiome. Next. So by which we can do them or utilize them for the basic studies. These are the first part I have explained about the gut bacteria. Second part is the endosymbionts. Normally the endosymbionts cannot be cultured, either the Wolbachia or Cardinium or Rickettsia. For all of them, we cannot culture them. We can only detect them using the specific molecular probe. So there is a technique by which we can use a specific primers by which we can characterize them. Second one is assessing the infection frequency. Say for example, if you are taking 100 individual, in how many of them they are present? Wolbachia, how many insects they are present? Rickettsia, how many? By which we can calculate only the infection frequency. The next method is using a qPCR, we can ascertain what is the relative abundance within the same insect, the proportion of the Cardinium or Rickettsia or the one, and we can assign the functional role also. Next slide. So this is a one study. We have characterized some of this gut bacteria. This is a study taken up by our colleague Dr. Kirti Sharma, and she has studied the gut bacterial symbionts from the fruit flies, and with the help of these fruit fly symbionts, they have prepared a kind of a consortium, and that consortium have been shown for their attractancy potential for the fruit flies. So this is one of the offshoot of the studies, not only studying the basic characteristics, how best we can use them for their pest management. In the case of fruit flies, we normally use the methyl-eugenol trap. They will attract only the male flies, whereas females as such, they will be giving the signals, and there are certain gut bacteria give the signals to them for attracting the females. So by using a repeated study, they have identified a consortium of symbionts in the fruit flies, and these fruit fly, after isolating them, Then they go for a volatile fraction extracted from these microbes, and they evaluated the attractancy potential of the different volatiles, and two prominent volatiles from the bacterial derived endosymbiont found to be attracting both males and females of the fruit flies. So this is one of the important area wherein the gut bacterial symbionts or the bacterial derived volatiles can be used for trapping of the pests. So this case it has been demonstrated in the fruit flies. Next. So there is a need for improving the attractants. The chemical use can be prepared. Similarly, in many different cases, even we can degrade we can remove the insecticide resistance by the populations. Similarly, in the case of termites and other insects, we can prepare the bait stations with the probiotic bacteria so that the subterranean insect can be attracted, and by using a proper bait, we can control them. These are the opportunities by which the gut bacteria can be, or the symbionts can be used for pest control. Next. Here I just want to show a small piece of our work wherein we demonstrated the association of the endosymbionts with the insecticide susceptibility or resistance. This is our paper wherein we found out the insecticide resistance status in the different genetic groups across India. Next. So we collected the different populations, and then we started isolating the endosymbiont from them and finding out are there any association or the difference in the resistant and susceptible population. And we found out that one particular Asia 2-7 population was more susceptible, whereas other population was resistant. Next. So we took the study for identifying the endosymbionts in the resistant and susceptible population, and found out in most of the susceptible cases the infection frequency of the Cordinium and Rickettsia were prevalent in the eye. Next. So this is only a small glimpses of the studies we have taken up, either for the structural characterization or functional characterizations, but there are opportunities are wide. Since India is one of the mega biodiversity country, there is a lot many scope for assessing the gut microbiome of the insects, either the insects of commerce like silkworm or honeybee, like my friend from Kashmir asked, there is a lot of opportunities for exploiting them for the beneficial insects. Similarly, Ganesh God also asked for their utility in honeybees. Same prospects we can also harness them for the lac insects, wherein these insects we can develop the probiotics and improve the productivity of the beneficial insects. Whereas in the case of pest insects, by designing the specific traps, we can trap the pest insects, or there are many different approaches for paratransgenesis by which we can apply for pest control as well. Next slide. So that's all. All these studies may not be possible without the funding agency support. I am fortunate enough to get the funding from the DBT, DST, NASF, and likewise many different organizations, our institute with all their supports, and the foremost is students and research scholars. With their contributions, it's all made possible, whatever little I have presented to you in these slides, in this one. By this way, I'm just thankful to all for providing funding support for conducting the studies, and also I thank you, the organizers, for giving me the opportunity, and the audience for their patient hearing for my lecture. Thank you all.
Okay.
Okay.
Next slide. The big thank you for me. Yes.
Okay.
Yes.
Okay.
Okay. Are there any queries from the audience side? The presentation has been, of course, very meaningful, very wonderful, and it was very crisp, in fact. And are there any questions, specific questions that you want to ask Dr. Subramanian, sir? The audience side, sir. Actually, there are a few questions in the chat box. If you can see there, otherwise I will read them out here.
Yes, sir.
Okay.
Can I ask the questions, please?
Somebody's
Probably Shivana queries us.
It is Shivana, sir.
Yeah.
Hello, Subramanian, sir.
Yes, please. Yes, please.
Thank you, sir, for your elaborative and very good information.
If you close the slide presentation, I could see them. I see you all. Just
Okay,
you can
you
close
can-
the presentation, Ganesh.
Yes, okay, sir.
Yeah,
Okay.
Yeah, we will close it.
Because I didn't see the chat boxes. Yes.
I'll-
Okay.
Fine, sir.
Fine.
Thank you for your elaborative presentation, sir.
Thank you.
And I have series of questions.
Yes, please.
The first question is, so I think we are also working on few of some endosymbiotic work in my laboratory.
Yes.
So we have done some insects, and yes, I am having one project related regarding the APID project from the DST, sir.
Yeah. Welcome.
Sir, actually we have evaluated some majority of some insects, even lepidopteran and other even apid species also. Generally, we found Bacillus, Staphylococcus, Enterococcus, and Astobacter, Pseudomonas. These kind of only groups we are finding in majority of the insects. So, is there any relation between the insects and only these kind of groups of the bacteria?
In most of the insects, Enterobacteriaceae is a family predominant. So whatever the
Yes,
insects
sir.
you are saying belonging to Bacillus and Enterobacteriaceae. These
Yes,
are the
sir.
two predominant groups, and third group is the Firmicutes. Only these
Yes.
three groups only we could come across in most of the phytophagous insects.
Yes, sir.
Yes.
You're right. And sir, actually regarding the Faggy system, I did not get the clear answer, sir. Faggy system, that monophagous and oligophagous and polyphagous. As per your study, it's fine. It's the proven that it is the oligophagous insect which harbors the more diversity of the microbes because it has to detoxify the cruciferin toxicants. Right. I totally endorse your statement, sir. But however, it should vary the difference between the polyphagy and the oligophagy and the monophagous. So my presumption that does it vary the load of microbes between the polyphagy, oligophagy and the monophagous?
Yeah, because monophagy, it will be feeding on only one repeated host year after year. So it will be adapted only limited host. So the microbes also only limited group of microbes, but distinct microbes that would be associated.
Yes, sir. But if that is the case, if that is the case,
polyphagous also maybe, because in our study polyphagous it is limited. We had taken up one polyphagous insect from only one particular host.
Yes, sir.
Say the same Helicobacter also, our studies is not elaborating holistically on the polyphagous. How the polyphagous diversity means, if you are taking the same Helicobacter from at least some 10 or 20 different hosts, still it would have given us some more information. Since with the scope of the study with one population, we could get only that much information.
Certainly, sir.
Yeah.
And even the selection of this host also, it will be driven by these symbionts. Do you agree with that, sir?
Yes, definitely.
Okay. And may I know, sir, from where you are outsourcing the sequencing even for metagenomics and others?
No, we rely on many different companies. Earlier, AgriGenome, Green Genome. So likewise, many of them are not consistent. In many cases, we'll get some of the results are not reliable. Many times we have to go for the resequencing. It happens so.
But we are also going for even for the AgriGenome, but supporting from the bioinformatics team is very difficult, sir.
Yeah, bioinformatics also. We developed our own bioinformatic analysis also by trial and error only. By having the contact with them of some of these, especially bioinformaticians who are trained in metagenome, we sought their help and improving our analysis. In the beginning, our analysis was very raw only. Now we are improving the analysis by taking the consultants and more experts so that it will throw more light on the data we are getting. Alright, whatever the outsourcing data the company are giving is very limited.
Yes, sir. I think I'm asking too much of questions. There's a last question from my side. Hello? Sir, can you hear me?
Yes, I could hear you. Proceed, please.
That is, sir, one study you have mentioned that there is a, and you have giving like mentioned that that is a diversity which mean different group, different areas you have collected the populations.
Yeah, in silkworm, Muga silkworm. Yeah.
Yes, for example, in different geographical locations, the microbial load is varies. So may I know what like drives them to be like varies in the among the microbes? What factors drive
These are the
them?
growing conditions of the temperature, adaptation.
Okay.
Because even within the northeast also wide variations will be there. What is there in Assam may not be the same in Arunachal Pradesh.
Yes, sir.
So both ecological conditions are in wherein they have to thrive.
Yes, sir. Thank you, sir. And small at the end, there is a small even for the root rub you have done that for northeast, no? That
Lepidiota mansueta.
Lepidiota mansueta, and we have like 3 species, sir.
Yeah.
We have 3 species which are very much like harboring, which is very much deadly dangerous for the arecanet. And all along the coastal system, I think if you combine these with these 3 species with those kind of root rubs, I think we can come out with good studies.
Yeah, good one. What is required is we also found out we need a more number of sampling. That is more important.
Yes.
One isolated sampling or one location or 1 year may not be sufficient. For a core microbiome analysis, first we have to have a wider sampling. Then only we could come to what are actually the core microbiome in the particular group of insects.
Sure.
Yes.
Thank you,
Because
sir.
once we know the core microbiome only, then we can go for elaborating them. Otherwise, whatever the sample studies, maybe the chance occurrence only for the particular year or for the particular population. So
Certainly,
irrespective
sir.
of the population or irrespective of the sampling, if they are getting the same from the same species of insects, then we can say them as the probably those core microbiome associated with the insects.
That means more number of samples and
Summer season, winter season, rainy season, likewise across the season, there will be the sampling efficiency you can increase.
Thank you, sir. See you soon at Delhi, sir.
Yeah, thank you. Whenever you come visit Delhi, we can interact more.
Yes, sir. Same lab, sir. I worked in same lab. Earlier,
Yeah.
Dr. B. Subramanian was there.
Okay, nice.
Okay. Are there any more questions from the audience side?
Sir, good evening, sir.
Yes. Uma.
Sir, excellent presentation, sir.
Thank
Sir,
you.
I have a question, sir. You were explained about the phytophagous insects, sir. Are there
Yeah.
any studies regarding the mycophagous insects, sir, like bark beetles or shorter borers?
Yeah, maybe I have not taken up studies. If you see the literature, especially Canadian literatures, they have worked so much on the xylophagy and the microphagy group of insects. But for them, you cannot expect the bacteria. Most of the microphagous insects, they are associated with the fungi or yeast.
Yes, sir.
So we have to do for those analysis with other, because our lab we are more conversant with the bacteria. We have taken up mostly with the group of insects we are conversant. So again we have to develop the expertise for different group of the microbes.
Okay, sir.
Yeah.
Thank you, sir.
Thank you.
There are a few questions. I don't know whether you are able to see them or not. So
Now you can anyway. I could see here.
I'll read them.
You can just click one by one. It's okay.
Okay. Can you see them? Sir? No.
Yeah, I can see. Just you can tell the questions. I can
Okay,
proceed.
fine, sir.
Because some of them have been already answered also.
Okay. Vijay Sairam Kumar.
Vijay Sairam. Yeah.
He has a question like wherein he says like any specific work on the gut microbiome analysis of a diptera done in India. That's a question that he has.
Are you able to culture them on any medium sans insect gut homogenate? That is what they are. I said only a limited proportion can be culturable. So that's why they are called as a culturable. So by taking the gut homogenate, we will be trying with the different media, because it's not a one single media we will be using. We will be using assorted growth conditions. So we will be pooling them all in the first stage, and then we can go for the isolation. The second one is, some of them are the obligate one, they cannot be culturable. They are called, we have to detect them only using the culture dependent approach, independent approach by the metagenomic analysis. Are you convinced, Vijay Shairam?
Of course, I don't know whether he's there.
Yeah, okay.
Yeah, okay.
Only bacterial symbionts are there. No,
Yeah.
many microbes are there. Our studies are focused only with the bacterial symbionts. Besides, there are flagellates or many different group of microbes are present. It varies with the group of insects. If you want to work on termites, specifically you have to focus on the flagellates and other group of organisms. Similarly, in some microphagy insects, most of the fungi are associated with the insects. Then very nice informative. These are the routine ones. Fruit fly, Girish.
Yeah, Girish.
Yeah, Girish.
Yeah. In the fruit fly gut attraction, that is what is the principal component which is responsible for attraction. We have done limited studies only, Girish. We have not taken up only all. We have taken only 3-4 volatiles from the few limited studies. This studies is only early stage only. We have taken up some of the identified 3-4 predominant endosymbiont associated with the reproductive tissues of them. We found out ammonia and 3-4, I don't have the exact, I don't remember those compounds also. Like they have tentatively labeled the component volatile 1, volatile 2. It's only very basic studies we have started. Still we had to go a long way in identifying the bacterial derived volatiles. But many papers are there, especially for fruit flies. Bacterial derived volatiles, excellent papers are there, especially they are done in Mediterranean fruit flies. A number of volatile compounds have been identified and they have characterized.
I have a small submission, sir.
Anything else, please, Ganesh? Girish, is it? Okay, please.
Did you have some study? Did you conduct some studies on Metarizum or Bavaria?
Nizar.
Nisa.
Yes, sir.
Yeah. Please proceed.
Sir, my submission is that did you have some studies on Metarizum or this Bavaria bassiana?
Yeah, I just showed on some passing information in a slide. We tried to prove the role of gut bacteria, especially Streptomyces gnocchi, that was an actinobacteria isolated from the silkworm. We tried to inhibit the spore germination of Bavaria bassiana and Metarizum anisopleae and tried to prove that these microbes may be in the inhibition of the fungal, especially the spore germination of these two fungi. But we have not taken any studies on the pathogenesis of these, because our lab mostly focus on non-pathogenic bacteria. We have not taken up anything on the entomopathogens.
Okay, sir. Thank you, sir.
Well, if there are
no more questions.
Hello, sir. Hello, sir.
Nagu, please.
Hello, sir. Good morning. I am from GKVK, sir.
Yes, please, welcome.
Sir, is there any possibility that endophytes become endosymbionts, sir?
Yeah, that is an entirely different area of domain. Because during the course of the evolution, some of these microbes have resided in the remain as an endophyte. Especially the sucking pests, they take up some of this endophyte also in during their phloem feeding, and they may reside. There are some circumstantial evidences are there in many different groups of organisms. We have to conduct a systematic studies only whether it is an endophyte, endosphere derived microbe or the insect. But there are possibilities are there, especially in sucking insects.
Okay, sir. Thank you.
Okay.
Probably
it seems like the people, the participants have completed asking their questions. So, sir, I'm very thankful to you, sir, Dr. Subramanian, sir. You started with the importance of structural and functional symbionts, and wherein you said for the sucking pests and the chewing pests, sucking insect pests and the chewing insect pests that I'm talking about, they will have the different biochemical milieu inside, and obviously the microbial, the fauna that will be there inside will be a totally different one. And this quest, that reason as to why this has been started is because the morphological
the techniques,
morphological techniques, they have the limited resolving power for the distinguishing the microbes. That's why you have, this has been started like culture dependent and molecular techniques, like independent and the molecular techniques you have started there in your state. You talked about the 16S rDNA primer, denaturing, gradient gel, electrophoresis, and next generation sequencing, and this has several artifacts like it will have a significant impact on the digestion, thermal tolerance, and decontamination of the toxins in the insect system. And there are, you also touched upon the factors that actually influence the colonization of the microbes inside the gut, so which will purely depend on the pH and the oxygen presence or absence and the redox potential. And the guide to microbial, the bacterial groups also you talked about, adaptive advantage to the population it gives, and the detoxification it helps in, and different types of the bacteria associated with the different insects, and also you talked about, and you gave an example about the microbial profile of the devium that you had done the research on, and healthy and diseased insects, because we didn't know, in fact, like, the healthy and the diseased insects, so they both of them have a different microbial groups. I mean, like, the situation will be totally different in the gut system when they are diseased, when the insects are healthy. And you talked about the nutritional symbiosis